for giving me the opportunity to present our start at this program. My name is Yukari Egashira. I'm working at Chiba University in Japan. Chiba University located in Matsudo City is very close to Tokyo. My specialty is nutrition chemistry, and one of my research is the study on the function of dietary fiber. Today, I will talk about the novel functions of dietary fiber and the involvement of gut microbiota. This slide shows contents of this presentation. First, I will talk about the effect of dietary fiber on obesity related to metabolic disease in human and mice. Second, I will talk about anti-inflammatory effect of dietary fiber. World Health Organization, WHO, reported more than 33% of the world adult population are overweight or obese. Obesity leads to the risk of inducing diabetes, hypertension, and dyslipidemia. These diseases are risk factor for atherosclerosis. A progression of atherosclerosis leads to ischemic heart disease or stroke. So the progression of these diseases are top 2 in the world cause of death in 2015 and 2020. So it is important to prevent obesity causing atherosclerosis. This graph shows the change in dietary habit of Japanese. After World War II, Japanese people's dietary habits have changed dramatically. In particular, the intake of cereals, rice, and potatoes has decreased significantly. Meanwhile, consumption of milk, fish, and meat and egg has risen greatly. So obesity is the major risk factor for diabetes, hypertension, and metabolic syndrome. So there are various factors that contribute to the development of obesity, including genetics, low level of physical activity, poor diet, and healthy behaviors. So recently, many Japanese people eat much fatty and other fatty and sweet food and do low exercise. So genetic environment factor and food lifestyle lead to obesity. Here is the top 3 list of the cause of death in Japan in 2014 and 2022. The Japan's Ministry of Health, Labor, and Welfare reports every year. Japan's 3 major causes of death for year have been cancer, heart disease, pneumonia, cerebrovascular disease. The cause of death in 2022 are also unchanged from those in 2014. If we added the rate for heart disease and cardiovascular disease, it's almost the same rate for cancer. That is top 1 cause of death in Japan. So metabolic syndrome is the name of the name for a group of risk factors that raises human risk of heart disease and other health problems such as diabetes and stroke. So risk for heart disease, diabetes, and stroke increases the number of metabolic risk factors. So risk of having metabolic syndrome is closely related to overweight and obesity and the lack of physical activity. The prudent diet, it's a healthy diet, is considered healthy and is characterized by higher intake of vegetables, fruits, legumes, whole grain, fish, and poultry. Compared to prudent diet, Western diet is characterized by abundant red meat, fat, and refined carbohydrates. The Western diet contributes to increase the risk of non-communicable disease, especially gastrointestinal disease and metabolic disease, which are partially attributed to the deficiency of dietary fiber in the Western diet. Dietary fiber is a widely recognized nutrient for human health. Dietary fiber is that of plant materials in the diet which is resistant to enzymatic digestion, which include cellulose, non-cellulosic polysaccharides such as hemicellulose, pectic substance, gum, and non-carbohydrate component lignin and resistant starch. The diet rich in fibers such as cereals, nuts, fruits, and vegetables have a positive effect on health side. And this consumption has been related to disease, decrease incidental of several diseases. Dietary fiber can be used in various functional foods like bakery, drink, beverage, and meat products. This slide shows the relationship between dietary fiber intake and coronary heart disease, CHD, death. This study was conducted in pool analysis. Pool analysis is a statistical technique for combining the result of multiple studies. This pool analysis combined 10 cohort studies. Cohort studies are a type of medical research used to investigate the cause of disease and to establish link between risk factor and health outcomes. Cohort studies are important to research design. It is a type of non-experimental or observational study design. The term of cohort refers to a group of people who have been included in study by an event that is based on the definition decided by researchers. For example, a cohort of people born in Indonesia in the year of 2023. This will be called birth cohort. The vertical axis shows CHD mortality rate, and the horizontal axis shows the fiber intake. When dietary fiber intake is between 18 grams and 20 grams, the relative risk of CHD death is 1. However, the mortality rate increases 30% when the dietary intake is 12 grams or less. On the other hand, it decreased by 20% when the dietary fiber intake more than 25 grams. So it is important to prevent CHD death to eat much dietary fiber. This slide shows the relationship between dietary fiber intake and blood LDL cholesterol level. This study was conducted in meta-analysis. Meta-analysis is investigators analyze the original individual level data from each study. The method of analysis is one of the most reliable studies. So also reported in human consuming a normal diet, psyllium, one of the dietary fiber intake had a total cholesterol lowering and LDL cholesterol lowering effect. Brown reported water-soluble dietary fiber suppress blood total cholesterol and LDL cholesterol. So dietary fiber consumption, prevent cholesterol, improve the cholesterol metabolism. This slide shows association between dietary fiber and type 2 diabetes mellitus in cohort. Dietary fiber, especially whole grain and or cereals intake suppress the onset of type 2 diabetes. So dietary fiber is useful to prevent diabetes mellitus onset in human study. So many epidemiological studies show that there was inverse association between dietary fiber intake and obesity-related metabolic disease. Next, I will talk about the effect of dietary fiber on obesity and its mechanisms. Animal experiments make it possible to analyze mechanisms that cannot be elucidated in human research. This study was conducted in my laboratory. These animal experiments were approved by the Chiba University's Animal Experimental Ethics Committee. The objective. This is psyllium. Psyllium has been cholesterol and triacylglycerol lowering effect in many animal and human studies. However, little information is available on the effect of psyllium treatment on activating the energy metabolism of muscle. In this study, we demonstrated whether the intake of psyllium activates energy consumption in the skeletal muscle using DNA microarrays. We use these black mice. Mice were fed a standard normal fat diet and high fat diet containing 40% fat in the diet. This is 64% energy from fat diet. Normal is only 16 energy from fat diet. And high fat diet plus 5% psyllium. This is 64% energy from fat diet. So the calories is same high fat diet and 5% psyllium group. So the mice ate this diet for 10 weeks. Plasma cholesterol, triacylglycerol, and blood glucose level was measured. And liver and skeletal muscle, the total RNA was extracted from this tissue. And then we done DNA microarrays was performed using Unpad instrument system. This Unpad system provided by Mitsubishi Rayon Corporation. A DNA microarray, also commonly known as DNA chip, is a collection of microscopic DNA spots attached to a solid surface. DNA microarray is an important tool. DNA microarray is an effective tool in transcriptomics that helps us in studying and analyze the mRNA expression of almost every gene present in an organism. The method is a basic protocol for DNA microarrays as follows. Locate and purify mRNA from sample of interest. Reverse transcribe and label the mRNA, hybridize the label target to the microarray, scan the microarray, and quantitate the signal. Recently, RNA-seq popular. RNA-seq can comprehensively examine gene expression level and perform a detailed transcriptomic analysis. However, this system is better to quantity, what amount we can measure the amount. So this slide shows the result, body weight gain and adipose tissue. Blue color shows the normal diet. Red color shows the high fat diet. Green color shows the high fat diet plus serum. So 2 weeks later, serum group decreased body weight gain. 2 weeks later, 5 weeks later, 10 weeks later, serum suppress the body weight gain. And this is the white adipose tissue weight. Epididymal fat, this is around fat around the testes. And retroperitoneal fat. And mesenteric fat, this is around the small fat around the small intestine. And subcutaneous fat. So this white adipose tissue decreased eating serum compared to high fat diet. 2 weeks, 5 weeks, 10 weeks. So this slide shows that serum has anti-obesity effect. This slide shows blood lipid and glucose level. 2 weeks later, serum suppressed triacylglycerol fat. And 5 weeks later, serum suppressed total cholesterol. And 10 weeks later, serum suppressed triacylglycerol, total glycerol, and blood glucose. So serum significantly decreased the plasma total cholesterol, triacylglycerol, and blood glucose level. So serum shows improves the lipid and glucose metabolism. This slide shows gene that change significantly in the liver and skeletal muscle. This is normal diet, high fat diet plus serum. High fat diet increase, green color shows decreased gene expression. Red color shows increase this kind of gene expression. So gene expression in high fat diet serum decrease these genes and increase the genes. On the other hand, skeletal muscle, high fat diet serum change these gene expression. We explain in more detail. This is the liver. Please show G6PC. G6PC is glucose 6-phosphatase, plays an important role for providing glucose in gluconeogenesis pathway. The reaction is to remove phosphate from glucose 6P. G6P is present in the liver and kidney. In celiac-treated mice, plasma glucose level decreased. Therefore, gluconeogenesis pathway accelerates. G6PC increase. And please show the HMGC. HMGC is a key gene for cholesterol synthesis. Blood cholesterol decreased by feeding serum. Therefore, cholesterol synthesis pathway accelerated and HMCA increased in serum group. So homeostasis is working in glucogenesis pathway and cholesterol synthesis pathway. This is gene expression cholesterol bile acid metabolism. These gene are cholesterol and bile acid transport protein, and CRP and NOS1 are inflammation-related proteins. So serum decrease this gene expression and also suppress this gene expression. So gene expression involving cholesterol, bile acid transport, and inflammation were downregulated in the serum group. This slide shows the serum suppress the transport system of cholesterol and suppress the inflammation. Next slide is the skeletal muscle. IL-12, IL-17, IL-18, and NOS3 are involved in inflammation. These gene expression in skeletal muscle are significantly reduced in the 5-part plus serum group. So serum may have anti-inflammatory effects in high-fat diet condition. So this is a skeletal muscle, and these genes are involved in fatty acid oxidation and LPL lipoprotein lipase, lipolytic enzymes that break down triacylglycerol. CD33 is involved in lipid transport. So increase this gene, so serum stimulate lipid transport and fatty acid oxidation in muscle of high-fat diet fed mice. So conclusion, serum stimulate lipid transport and fatty acid oxidation in the muscle in high-fat diet fed mice. So this phenomenon is partly attributed to the ameliorate lipid metabolism and obesity. So conclusion, this experiment suggests that dietary serum can enhance the transfer of excess fatty acid from the liver to the skeletal muscle and promote lipid consumption in the skeletal muscle. Serum contributes to the consumption of lipid of muscle, and this phenomenon is partly attributed to ameliorate lipid metabolism and obesity. Next slide, I will introduce the effect of elderly mushroom dietary fiber on obesity and gut microbiota in mice fed a high-fat diet. Do you know this mushroom? Soluble dietary fiber have been reported to yield a variety of health-promoting activities. Dietary fiber has significant implications that gastrointestinal health by regulating the gut microbiota. Many studies show that the physiological function of different types of dietary fiber depend on their physiochemical characteristics, especially solubility and viscosity are important. Compared with insoluble dietary fiber, soluble dietary fiber can be easily accessed and metabolized by fiber-degrading microorganisms in the intestine and produce a series of beneficial and functional metabolites. Barley and yeast fiber beta-glucan have reported anti-obesity effects, cholesterol-lowering effect, fatty liver improving action in mice and humans. However, little information is available of effect of eringi. European people say king oyster mushroom fiber on obesity. Eringi is the largest species in the oyster mushroom genus. The samples used in the experiment is soluble in hot water and precipitated in ethanol among polysaccharides contained in LNG and contain a lot of beta-1,3,1,6 glucan. In this study, we examined the anti-obesity effect of water-soluble glucan derived from LNG using high-fat diet-induced obese mouse. Aim. In this study, we investigate the effect of LNG-soluble dietary fiber on obesity and gut microbiota in mice fed high-fat diet. This slide shows the scheme for preparation of soluble dietary fiber from LNG. LNG was obtained from Hokuto Corporation in Japan. Soluble dietary fiber was extracted with 2 volumes of hot water from raw LNG. It was boiled for 1 hour. After cooling, we added 4 volumes of ethanol to extraction at room temperature. After overnight, I collected precipitate using filtration and then dried at 80 degrees overnight out powder. The LNG powder sample contained 90% dietary fiber, 2% protein. The majority of constituent sugar was glucose. LNG fiber contains a lot of beta-1,3,1,6 glucan, and average molecular weight is 740 kilodalton. So this slide shows annual experimental design. Mice were randomly divided to 4 groups. Each mice fed the normal diet, high-fat diet plus 1% LNG fiber, high-fat diet plus 5% LNG powder. High-fat diet group are almost the same energy from fat. And after 6 weeks, the mice were killed and tissue was collected. Adipose tissue were weight and serum total cholesterol, TG, HDL cholesterol, LDL cholesterol level were measured. This slide shows the body weight gain and white fatty weight at the dissection. Blue color normal diet, gray color high-fat diet, orange color show high-fat diet plus 1% LNG fiber. Dark orange color shows high-fat diet plus 5% LNG fiber. So this graph shows 5% LNG powder decrease the body weight gain compared to high-fat diet. This slide the white adipose tissue and almost white adipose tissue weight is decreased by feeding high 5% LNG fiber. These results indicate that LNG fiber intake suppresses visceral fat accumulation and obesity. So this slide shows the biochemical parameters in blood. There were no significant difference in the serum glucose, TG, nephra among the high-fat diet group. However, serum total cholesterol and LDL cholesterol level decreased in the dose-dependent manner with LNG fiber intake. So LNG fiber intake improves cholesterol metabolism in high-fat diet. Next, in order to investigate the influence of LNG fiber on bile acid excretion, we measure bile acid in feces. LNG fiber intake increases fecal bile acid in a dose-dependent manner compared with high-fat diet. These results suggest the excretion of bile acid was promoted by LNG fiber ingestion. And we measure fat content in feces, almost the same result. So bile acid synthesis from cholesterol in the liver concentrated in gallbladder and secreted into the duodenum as bile. It then forms micelle in the gastrointestinal tract and helps absorb fat in the food. About 95% bile is then reabsorbed in the ileum and again secreted as a bile. In this experiment, it was suggested the excretion of bile acid was promoted because the amount of fecal bile acid increased due to the ingestion of water-soluble dietary glucose. In liver, cholesterol metabolism is controlled by cholesterol synthesis key enzyme HMG-CoA reductase and incorporation of blood LDL cholesterol by LDL receptor and catabolism of cholesterol to bile acid by CYP7A alpha. Moreover, HMG-CoA reductase and LDL receptor are regulated by transcription factor SREB2, and CYP7 alpha is regulated transcriptional factor LX alpha. So we measured this gene expression. And this is the result. A psyllium feeding bile acid exclusion was increased. So bile acid synthesis from cholesterol, therefore liver cholesterol level was decreased and to produce bile acid. As a result, decrease in cholesterol in the liver, SRBT2, which sense of decrease in cholesterol, was activated, and expression of its target gene, LDL receptor and HMG-CoA reductase, was increased. The increase of uptake of blood LDL cholesterol into the liver, resulting in decrease in blood LDL cholesterol. So this slide shows the change in gut microbiota at pheromone level. The relative abundance of green color bacteroidetes. A new name is Microbiota. And orange color actinobacteria, new name is Actinomycelotota, were lower in the high fat control group and eating fiber group compared to STD normal diet group. And relative abundance of firmicutes, it is known as obese microbiota. Firmicutes and Probacteria, orange color, were higher in the high fat control and eating group. So, pheromone level, there is no difference between high fat diet and psyllium groups. However, we focus on genus level, changing gut microbiota genus level, high fat diet, and eating fiber group are different. This slide shows the change in gut microbiota at genus level. At the genus level, there were differences in the gut microbiota among the groups. This is a change in gut microbiota at the genus level. Parabacteroidetes is involved in the suppression of enteritis, small intestine inflammation. And lactobacillus is lactic acid producing bacteria. Roseburia is butyric acid producing bacteria. So, Parabacteroides, Erini fiber includes Parabacteroides, and Lactococcus is lactic acid producing bacteria. Standard group and elite group increase. So this means produce lactic acid. And Roseburia and Lactobacillus are butyrate producing bacteria. Enemy fiber tend to decrease these bacteria. Clostridium is butyrate producing bacteria. So enemy fiber tend to increase clostridium compared with high fat diet. So these results suggest that gut microbiota composition change with enemy fiber intake. So ingestion of dietary fiber changes the composition of intestinal bacteria. Intestinal bacteria ferment or break down dietary fiber to produce various short-chain butyrate, propionate, and acetic acid. One of these, butyric acid butyrate, acts on gene and promotes the differentiation of regulatory T cells. T cells maintain immune homeostasis and suppress inflammation. And butyrate activate PPAR gamma. PPAR gamma causes insulin sensitization and enhance glucose metabolism and anti-inflammatory action. It is a master regulator of adipocyte differentiation, yet promotes insulin sensitivity. And dietary fiber leads to satiety. And butyrate causes satiety. So bind to dietary fiber induced short-chain acid binding of peptide YY. Binding of peptide YY to brain receptor decrease appetite and make people or animals feel full after eating. So, and more about suppress energy intake, TNL have decrease and decrease adipogenesis and some increase thermogenesis and lipid deposition. So short chain fatty acid a lot of function, especially energy metabolism and inflammation. And dietary fiber change the intestinal microbiota and produce short-chain fatty acids such as butyrate, propionate, acetic acid, and this change insulin sensitivity and decrease fat activation and increase energy consumption in muscle and increase insulin sensitivity in liver and muscle via GPR47 receptor. So short chain acid affect the glucose, lipid metabolism and inflammation. So conclusion, in this experiment, it was suggested identify the bile acid excretion and promote by ingestion of LNP fibers. So catabolism, cholesterol from cholesterol to bile acid increased. As a result, stored cholesterol in the liver decreased. And SRBP was activated and target gene LDL receptor and gene receptors decrease. Result, LDL cholesterol in the liver decreased. That's a mechanism. So in this study, it was suggested that cholesterol catabolism in the liver and LDL cholesterol uptake was promoted by ingestion of LNP fiber, which lead to decrease in human total cholesterol and LDL cholesterol levels. In addition, suppression, visceral fat accumulation, and improvement in fair glucose tolerance were observed by ingestion of LNP fiber. Increase of adiponectin may be involving ameliorated glucose tolerance. Moreover, LNP fiber intake increased lactic acid producing bacteria and butyric acid producing bacteria in gut. And GPR47 expression is increased in visceral fat accumulation, fat increase in energy LNP fiber intake. So short chain fatty acid may be involved in anti-obesity effect. This is the conclusion. Finally, I talk about the effect of dietary fiber of inflammation. BioBran is a rice-bran dietary fiber, water-soluble dietary fiber, named MGN3, is a partial hydrolyzed product obtained from treating water-soluble dietary fiber from rice bran with enzyme from Bacillomyces, one of bacteria. So rice bran fiber have various physiological actions such as anti-tumor and immunomodulatory effect. We used hepatitis model. That was induced hepatitis induced by D-galactosamine. D-galactosamine induced liver damage in the rat. Since histological and biochemical findings of this galactosamine-induced liver damage are very similar to those human hepatitis B. This is the mechanism why galactosamine-induced hepatitis. Galactosamine injection activate mast cell. Mast cell release histamine. Histamine open the gut-tight junction. So bacteria in the intestine are located to blood. So blood bacteria activated Kupffer cells in the liver, and Kupffer cells release inflammatory cytokine, TNF-alpha, IL-18, and on the other hand, galactosamine metabolized in the liver and galactosamine metabolized to UDP galactosamine. So UDP reduction lead to malfunction protein synthesis and mRNA expression synthesis. So this leads to apoptosis and necrosis. So both mechanisms lead to hepatitis B. So in this study, we investigate the inhibitory effect of rice bran hemicellulose on galactosamine-induced liver damage. Rice bran was administered intraperitoneally or orally to rats. Then 1 hour later, galactosamine was administered intraperitoneally, and dissection was conducted for 24 hours after galactosamine administration, and serum transaminase activity was measured. This is a result. Effect of rice bran hemicellulose on galactosamine hepatitis. Serum transaminase activity is induced by hepatitis. This is a normal rat. That is bigger than mice, but looks very similar. So yellow color shows GOT, and another name AST or ALT. Green color is GPP. However, galactosamine treatment increases the GOT and GPP. Serum trans activity increase means induce hepatitis. However, rice bran hemicellulose suppress serum transaminase. So this means rice bran eating ameliorates the hepatitis induced by galactosamine. And ILT is known, it's related to hepatitis. So compared to normal diet, galactosamine-induced IL-18 mRNA. However, rice bran suppress IL-18 mRNA expression, and this is a serum IL-18 concentration. IL-18 concentration was suppressed by rice bran hemicellulose. IL-18 overexpression mice cause hepatitis, and IL-18 knock mice, if got the degalactosamine treatment, they didn't become hepatitis. So IL-18 is important and concerning about hepatitis. So next, we hypothesis of hepatic suppression mechanism by rice bran hemicellulose. Rice bran hemicellulose prevent translocation from gut to blood, and bacteria activate the bacteria, especially LPS, bind CD14 and activate toll-like receptor 4 and activate the MAP kinase and NF-kappa B system. These are inflammatory factors and induce IL-18 mRNA and mature IL-18 cause hepatitis. So rice bran hemicellulose prevent this or this or this. We conducted the experiment. This is gene expression of CD14. Normal diet is low level of CD14. However, control increase, fiber decrease. This is toll-like receptor mRNA levels. Normal is low. Hepatitis increase. However, fiber is low. And then we measure the expression of I-kappa B alpha. Standard is dark and hepatitis low. However, fiber recovered the protein expression. So this is MAP kinase protein expression in lab. We measure especially JNK phosphorylation is a target by dietary fiber. So conclusion, MGN3, rice bran water soluble dietary fiber prevent this and this and this. Decrease CD14, suppress bind to LPS and LDL toll-like receptor for activation. This causes reduced nuclear transfer of NF-kappa B and MAP kinase, such as P38 NK phosphorylation. IL-18 mRNA was decreased by inhibiting nuclear translocation of NF-kappa B. So as a result, it was possible to reduce IL-18 release into the blood and prevent liver damage. So today I talked about the effect of indigestible polysaccharide on obesity-related metabolic disease and inflammation. WHO reported more than 33% of the world adult population are overweight or obese. Obesity leads to the risk of inducing diabetes, hypertension, or dyslipidemia. These diseases are risk factors for atherosclerosis. A progression of atherosclerosis leads to ischemic heart disease or stroke. These diseases are top 2 in the world cause of death in 2020. So it is important to prevent obesity. Dietary fiber prevent obesity in human and mice. Many human studies show that there are inverse association between dietary fiber intake and obesity-related metabolic disease. So it is important to prevent obesity causing atherosclerosis. So let's eat dietary fiber every day. And recently, research on the brain gut axis has been attracting attention. There is a report in 2020 that when diabetes mice induced by high-fat diet eat beta-glucan from barley. Eating beta-glucan improved the intestinal environment and suppressed brain inflammation and improved cognitive function. Because high beta-glucan strengthens intestinal barrier function, intestinal defense, and beta-glucan suppress bacteria LPS flowing from intestine to blood. This phenomenon improves cognitive function in diabetes mice. However, I'm interested in this experiment. However, further research is needed to know the relationship between dietary fiber intake and brain function. My laboratory study was conducted by many students, including Dr. Dian Kuniati. So, indeed, Mr. Kash and Mr. Togawa conducted serum experiment, and Mr. Nakahara conducted Ering study, and Dr. Surina conducted rice bran study with me. So today I introduced the dietary fiber function and mechanism. Thank you very much for your attention. That's all.
Thank you very much, Sensei, for your interactive and interesting presentation regarding the dietary fiber and its impact to the health. And I hope we can have a good discussion in Q&A session, Sensei. Okay, now we move to the question and answer session. And I'd like to invite the participants to give the question to our speaker. And the participants can ask directly to our speaker by raise your hand, or you can type your question also by the chat box. Okay, Sensei. We have lot of questions, Sensei.
Oh, really? I check.
Okay, maybe
Oh,
I
oh.
will read it one by one. You can also see in your chat box, Sensei. Okay, the first question is coming from Miss Indira. In your opinion, is consuming drinks that contain soluble dietary fiber every day safe for our health? And then which one is better, to consume soluble dietary fiber without processing, or processing it first? Maybe you can give your response one by one, Sensei.
Yes. Drinks, consume drinks that contain soluble dietary fiber every day. Safe. I should, please change. Sorry. Depend on the drinks. For example, recently some drink added polysaccharide, dextrose, and Otsuka company in Japan produce 5 mini. 5 mini, it contains 7 gram dietary fiber, 100 milliliter. So first we drink for 1 hour. And some people cause diarrhea, but after 1 week later, the microbiota adaptation cause. So it's safe. And recently, there is resistant dextrine developed. Cola with resistant dextrine is very popular and hit in Japan. However, I think dietary fiber is many cereal materials, and fruits contain much dietary fiber. So I think get dietary fiber from natural food, for example, seaweed or fruits. And vegetable is better, I think. However, safety test was checked in my country, and safety is okay. It's provided to people. So every day is sometimes you can drink, you can use drink is better, I think.
Okay, Sensei, thank you for your response for the first question. Mrs. Indira, is it clear for you? Okay, maybe we move to the next question, Sensei. We have a question. The question is coming from Arifin. Arifin. She has 2 questions. First, could you explain the mechanism or how the natural resources that you introduced, such as psyllium and king oyster mushroom, inhibit or reduce the risk of disease associated with obesity? And the second question is, what is the optimum dose of psyllium and oyster mushroom in a daily diet with a significant effect, Sensei?
Serum in generally, 25 gram dietary fiber is recommended. But serum is very expensive. So if it's get every day is very, very expensive. However, 25 is more. What dietary fiber is water-soluble dietary fiber and insoluble dietary fiber. In general, 20% are soluble fiber and 80% insoluble fiber is better. So, but serum is water-soluble fiber. So maybe 5 gram is better, but very expensive. So it's easy to get natural food. And oyster mushroom diet. In this experiment, oyster mushroom purifies only dietary fiber. So it's very but oyster mushroom contain not so much dietary fiber. If we prevent obesity, supplement, concentrated and supplement is available. But I think not only this serum or LNG fiber, but every plant material contain dietary fiber. So I think there I recommend several kind of dietary fiber and several kind of vegetable or cereals is better. Is it okay?
Yes, thank you, Sensei, for your response. How about the response, Mrs. Arifin, Henny? Is it clear for you? Or you have any additional question? Maybe clear, Sensei. Okay, let's move to the next question. Yeah, the question is coming from Miss Vera. She asked about what are the challenges and limitations of this research and how the product development for this topic. Sensei.
What are the challenge and limitation of this research? Limitation, challenge and limitation of this research. How is product development to the topic? How is the product development is a challenge and limitation of this research? This research is shows the mechanism, detailed mechanism in gene expression, and limitation is human experiment is more important. Animal experiment is useful to check the mechanism. So human experiment and animal experiment is not almost the same. So if we get the good result from animal experiment, then try to human experiment, not only cohort experiment, but also the intervention study or gave the some kind of dietary fiber to humans. So I think doctor attend to our project is better.
Okay, Sensei. Thank you very much for your response. So it means that this study needs the further research to the human. The next question is coming from Mr. Agus. He asked about the dietary fiber can reduce the risk of obesity, which can lead to narrowing of blood vessels. If someone switch food consumption from glucose-containing foods to dietary fiber, are there any side effects caused by the consuming dietary fiber? And where we know the body needs glucose intake to become a source of energy.
Depend on the amount. Depend on the amount. So a large amount of dietary fiber cause sometimes side effect, but in generally, it's difficult to get a large amount of dietary fiber from our daily food. So, in vitro experiment, dietary fiber combines some kind of mineral and water-soluble dietary fiber cause diarrhea, a large amount of intake. However, in our daily food, adverse effect or there is a few reports adverse effect in vivo experiment and in human experiment.
Okay, Sensei. Thank you very much for your response. Mr. Agus, how is the response? Is it clear for you? Okay, maybe we move to the last question, Sensei. From Mr. Awali. The question is, it's very interesting topic about eringi and having high productivity as a cultivated mushroom, and it can also have a positive effect to as an anti-obesity. But he curious if we consume eringi at an excessive dosage, could it have any negative effect to our metabolism?
We didn't measure the toxicity evaluation, so I don't have the data. But I think purified eringi fiber prevent obesity. I think so many people eat eringi every day. So I think many people eat mushroom, including eringi. So daily food intake is, I think, no problem. However, if you get a supplement from purified eringi fiber or another materials, purified fiber, more experimental data is needed. This is one of the just animal experiment. So in the future, try to do a human experiment and confirm the safety experiment evaluation.
Yes, Sensei. Thank you very much. Sensei, one more question, Sensei. Is it okay? Yeah. The question is coming from Mrs. Siti. And why are you studying obesity as your research topic? And because some people outside Japan think that Japanese people have no concern about obesity since most of them have an ideal body. Begitu for the
because
question.
yes, because recent Japanese people food lifestyle is changed. And previously, we eat a lot of potato or cereals, but recently, food style changed. Western Western style. So we eat FTIP. For example, I like McDonald's fried potato and hamburger. The taste is very good. And ice cream and cake. So, many people told me I look thin, but not my cholesterol level increase and blood level a little bit increase. So there is a relationship body weight gain and blood glucose and lipid cholesterol value. So, and obesity is very important problem in Japan. So, I focus on the obesity. And Japanese people, Japanese looks smart, but not so. Especially middle-aged old man is obesity and hypertension and high blood pressure and suddenly death. So it's to prevent atherosclerosis. So recently, many Japanese people concentrate on obesity research.
Important things is that everyone needs to maintain their body and health. It seems our question is no questions, no more questions, and we finally come to the end of this seminar session, Sensei. But before I close this seminar, I would like to take the conclusions from what you has presented, and there are some points highlighted that psyllium as a dietary fiber is beneficial to maintain health, and specifically can reduce the obesity, decrease the cholesterol, and decrease the glucose level in the blood. And the second is the dietary fiber can be an option to reduce the potential high-risk disease, that, for example, diabetic, cancer, and so on. And Sensei, before I close this session, on behalf of the committee, I'd like to deliver the certificate for you for your valuable contribution today. Sensei, thank you very much.
Thank you very much.
Okay. Okay. Again, I'd like to thanks to the Egashira Sensei to your contribution in this seminar session, and also to all the participants for very active participation. I'm Devi Maulida Rahmah, the moderator of this seminar today. The Master of Ceremony.
And also, we would like to thank Ma'am Devi for being our wonderful moderator in this session.
And thank you very much for Professor Yukari Egashira for giving such an informative and comprehensive presentation for the second part. As a token of appreciation,